Welcome back, last session of the day. We've heard a lot about, pathogens, their sequencing, and Milind's talk will hopefully integrate these perspectives which we had from the previous few days of ecology and evolution, and also, these variants and sequencing. So, Professor Milind Watve, it's a real honor that he could make it. He did his PhD actually on pathogen and parasite load very long ago when people were not thinking about these things at all in elephants. And subsequently, he worked initially at the Garware College, where he was nucleating, many groups of young students who are interested in ecology and evolution. And for some time, probably until about 10 years ago, many people who have any interest in ecology and evolution probably had somehow interacted with him and worked with him. So after that, he was at ICTS Pune, and it's really a pleasure to have him. Thanks for coming, Milind.
Thanks, Uma. Thanks to organizers, because my addition was a last-minute addition. Actually, I would have loved to be a participant and be here all the days, but somehow I didn't know about it. Let me add a little bit to my introduction, because the setup in which I work is very different. So 5 years ago, I decided to quit academia and work on my own, work with people. Okay, so the research continued. Research did not stop, but the model of doing research changed. Okay, so now I work completely as a citizen, as an individual. There is no organization, there is no funding, there is no PhD student, there is no fellowship, nothing. We work on a 100% voluntary basis. Anyone who is interested can join hands, and we work on something that interests us. Okay. Now, I decided to quit academia because I understood the limitations of academia and the current tendencies. Quite a number of times it has so happened that I talked with someone, we found that, oh, this is very interesting, let us join hands and start working on this. But the other fellow said, but then it is very unlikely for this idea to attract funding. Okay. So then with one professor in the University of Glasgow, we thought of idea, we thought of collaborating. He applied for, we jointly applied for funding. That funding did not come through. And that at stage, he gave up. I didn't. Okay, but he gave up on the idea. Now, this is very typically the mindset of academics today, that their research begins with getting funded, getting a PhD student, and the prospects of getting a good paper. Okay, so in contrast to that, now we work without any of these constraints. Okay, we work simply because something interests us. Okay, whether we will end up publishing that or not is entirely immaterial. As a result of this, in the last 5 years, as a group, we have published more than what my output in an institute was. Okay, so published more than 25 papers in the last 5 years, ranging, although we did not bother about impact factors or on, it ranged from impact factor 0 to 29 and all. And one of the pieces of work where my co-workers were farmers, some of them were illiterate, but with a little training, they maintained their own data. Okay, and that got an international award as well. Okay, so I'm trying to do research outside academia as a different model, and the strong point of this model is you really become a thinker. And that thinking is not constrained by whether you have a PhD student, whether you have funding, or whether you will get a high impact publication. Interest is enough. Okay, both necessary and sufficient, and nothing else is needed. Well, so incidentally, I will start with where the last speaker stopped. Okay, the last question that Farah asked. About the variants and the waves. Okay, and well, then how do we work? Okay, as I said that we have no funding, no fellowship, no PhDs, then how we work? A lot of science can be done without infrastructure, without anything. Okay, just you have a thinking mind, a questioning mind, a group of interested people for a number of reasons. There is an open lab out there. Okay, once you go out, there is a huge lab open. People is another lab that is open and anyone can experiment. And the third one is the good thing of today's science is there is huge amount of data in the public domain, which you can easily access. Okay, and with the same data, if you ask new questions, you get something new out of it. Okay, so mostly we work with public domain data, and there is a lot in the public domain data that is not yet interpreted. Okay, I will just give you a few examples. Fine. So this is simply how they picturize the different variants coming, and this data is there on that coherent.org website with a time resolution of 15 days. Okay, so all the sequences that were done in that given country for the last 15 days, they calculate the proportions. Okay, and what you see in the different colors are the proportions of different variants with time. Okay, so what are we going to do with it? Well, the question that we ask is whether the evolution of new variants is mutation limited or selection limited. Okay. Now this is a new question. People generally haven't asked this question. But what do you mean by limited? Okay. For people in research, this is, so you want to start one project and you need clearance from so many committees and departments. Okay, so you need funding, and funding sanction is not enough. It has to actually arrive, and that is completely unpredictable. Okay, so funding has to arrive. You need clearance from, there is an IBR, there is a ethics committee, then you might need clearance from some other ministry and so on, right? So you need so many clearances in order to start the project. Well, even if one of them is late, the whole thing is held up, right? There is no use others being fast. You go by the speed of the slowest, right? So the slowest is the limiting factor or the rate limiting factor, okay? And perhaps bureaucracy is the best example of rate limiting factor. Now, it is inevitable because you might have a task force to reduce government bureaucracy, but the task force will have a chairman and a committee and okay, so that doesn't work. Another metaphor that if you try to run by joining hands, then you end up running by the speed of the slowest. Okay, it is not the average speed, it is the speed of the slowest, right? So this is called limiting. And in any complex system, any complex process, it is extremely crucial to identify the limiting factor, right? Because even if others are all well and fast and quick, prompt, it doesn't work. The limiting one has, so any remedy has to be focused on the limiting factor, right? Now in evolution, well, two fundamental things that make evolution is mutation and selection, okay? Not as simple as it looks, because in mutation, again, there are details. In selection, there are so many complexities, but to make it simple, there are two factors. There is mutation and there is selection. And whether evolution is mutation limited or selection limited is a question that is highly underrated or highly assumed, but this question needs to be addressed very seriously. Okay. Typically, the Darwinian concept of evolution was selection limited. Darwin even did not know what mutation is, okay, or what is the mechanism of inheritance. Even in the later phases of evolutionary theory, the mainstream evolutionary ecologist would think of selection, would not think of mutation. There it is assumed that mutations will take place anyway. Okay, so the selection is something that matters. Interestingly, there are a few other examples of evolution where the thinking has been mutation centered. Okay. Now this one is a fairly clear example. In bacteria, when we work, the populations are huge. Okay, a single colony is 10 to the power 9, 10 organisms, and you have hundreds of colonies on the plate. And then mutations are bound to arise because the populations are large. Populations are greater than the reciprocal of mutation rate. Okay, so that is the condition. And if that is so, then the process is not mutation limited. It is selection limited. On the other hand, if your population is small, so the chances of mutation are small. Okay, but there is strong selection pressure, then it's a mutation limited process. In thinking about evolution of cancer, now I'm talking about somatic evolution of cancer. Cancer has to evolve every time within a body, okay, because there is a series of mutations and there is a selection accompanying these mutations. So cancer for a long time has been thought of as a mutation limited process. Okay. Now, very recently, about half a dozen papers, including 2 of ours, have actually tried to show that no, cancer is a selection limited process, not a mutation limited process. Okay. So, this is a fairly complex analysis. I'm not going to go into that, but you can only think of what is the number of stem cell divisions in a lifetime human. Okay, this far exceeds the mutation rate, right? Which means that cancer causing mutations must be happening in almost everybody during the lifetime. Everyone doesn't get cancer. This is because it's not only the mutation, what happens to the mutant is what is important. Whether the mutant survives, the mutant has to compete with normal cells. Okay, how the dynamics of that competition goes, okay, and whether the mutant survives and proliferates so that another mutation might happen when the first one has proliferated enough. Okay, so this is not a mutation limited process as people are thinking mostly in the field of cancer. People talk about mutation, hardly anyone talks about selection, but cancer is a selection limited process. Again, this is a fairly complex mathematical argument. I will not go into it for 2 reasons. Number 1, I don't want to bring much mathematics here. Number 2, I am very bad at mathematics. Okay. Okay. And then the question that I am going to address here is whether the evolution of new variants in the SARS-CoV-2 during the pandemic was mutation limited or selection limited. Okay, again here, the prevalent thinking is of being mutation limited. Now, although I will not go into any equations or any simulations or calculations, we can do some quick mental calculations or at least mental judgments, if not calculations. Fine. Viral populations are huge. Okay, within a single body, there are billions of virus particles at the peak of infection. And when there is a pandemic where thousands or even millions are infected, the net viral population is just tremendous. When do you think probability of mutation is highest? Very simply, and let us think about a given mutation. Okay, a mutation which you think gave rise to slightly oversimplified, it's not a single mutation, but at some time you think that a new variant arises. Okay, and we think that the new variant is perhaps more infectious, or for whatever reason, people believe that variants cause waves. Okay, that also we are going to examine, whether variants cause waves or waves cause variants or neither of the two. But when is the probability of mutation maximum? It is at the peak of a wave, right? Where the viral population is maximum likelihood that a given mutant will arise, okay, or a new variant will arise. Now, people believe that if a new variant evades the immunity to the prior variant, then that is reason enough to cause a new wave, right? So now, just put these things together. New variant can evade the immunity, and new variant arises at the top of the prior wave, right? If you simulate this, write a mathematical model and simulate this, then it happens that as the wave goes up, it will never come down, because when it is at the top, a new variant arises. Okay, it will start spreading because you believe that it evades the immunity to the prior variant, right? By the time this is at the top, a new variant is likely to arise. So if new variants evolve and if this evolution is mutation limited, then once an epidemic goes up, it will never come down. Okay, definitely this was not the picture, and which means that there is something wrong in our thinking. Okay, look at when did every new variant arise or first detected. Okay, arise and first detected, there is a slight difference, but when did it arise or when did it first detected? Almost all of them are not at the peak, but when the incidence was low. Okay, there is much more data. This is country-wise. I am showing just a few examples. Okay, so as we said, new variants don't seem to arise at the peak of the prior wave. Okay. They mostly arise when the disease is at a low incidence, only few like a low-level endemic-like state, and so on. Why does that happen? Okay, and if you look at the history of all variants, 90% of them have arisen when the infection was at a low level. Well, here it is the frequency distribution of time from the prior wave to the invasion of a new variant. Okay, and you will find that very little when the earlier wave was at the top. Okay, the peak is at about 100 days. So maximum variants have come up after about 100 days of the prior wave waning. Okay, so there is something to do with the prior wave. Let us look at the other side of it. Okay. Can you get this pattern of repeated waves without a new variant? Okay, and this is an yes.
I just want to clarify this. So you are saying that when the variant has been detected and when the infection level is very low, there is a buildup of 100 days or something you are saying
Yes.
till the waves comes up.
Yes.
But isn't that why would you not expect that? You will, right? Because it takes certain amount of time for that variant to infect certain number of people, and that also depends on the RO, how fast, how infectious the variant is, right? So I am not able to understand why would you expect it otherwise? I mean,
so why 100 days for it to start?
no, it's not a
infecting?
consistent thing. Some variants come very fast, some are very slow in build up to the wave. if you're causing it.
So we haven't come to the wave as yet.
I see.
Okay, so this is about when the variant arises. Okay.
Excuse me.
Yes.
So that previous few graphs are showing the waves for different countries. Can't this be the case that a new variant is coming from a different country and not really mutating and coming?
So there are two things. In this graph, sorry. So the one on the left, this depicts the origin, wherever it was first detected, whichever country.
Oh, I see.
Right? What you have on the right includes both, whether it has traveled from outside or originated there.
All right.
Okay. Okay. So this is the other side of the story. Is it possible that repeated waves happen even in the absence of new variants? Okay. Now, this is a completely theoretical model. Again, I will not go into the mathematics, but this differs from the classical models. The classical epidemiological models are called compartmental models, where they divide the population into categories like susceptible, infected, immune, may be removed, that is, they are removed from this process because they are either immune or dead. Okay, so these are called SIR models or compartmental models. What they do is their immune is immune. It's a binary variable. Either you are susceptible or immune. Okay, we made a different model in which we treated immunity as a continuous variable, not binary. See, that single change makes the model extremely complex. Okay, using binary variables is extremely simple. But we made immunity as a continuous variable, which is realistic. Immunity is not binary. All immunologists know that immunity is a continuous variable in terms of titer, in terms of memory cells. So immunity is a continuous variable. The models were not treating it as a continuous variable. Okay, the moment you treat immunity as a continuous variable, so many things, so many outcomes change. Okay, one of the changes is you get these repeated waves without a requirement for a new variant. Okay, and this is because the brown line, which is the mean population immunity, okay, goes up, and particularly if immunity is short-lived, then it comes down very fast, and after a while, when it crosses a threshold, the population is susceptible for a new wave. Yes.
How did you represent the immunity in this?
So, if you have to make immunity as a continuous variable, the model changes completely. Okay, from compartmental model, it becomes an agent-based model where every individual in the population has a level of immunity which changes according to the history, whether there was a prior infection, whether there is vaccination, and whether there are subclinical exposures. With subclinical exposures, immunity is known to go up slightly. Okay, and then in the absence of these exposures, immunity starts declining as well. Right? All these processes can be brought into the model, but now this model becomes an agent-based model.
But if you just put an arrow from the recovered population back to the S, that way, if you model it, you can still see multiple peaks as a function of time.
So, that is highly conditional. That is one. There is another difference. I will come to that. Okay. So,
I thought that the compartment models are essentially representing a population. So
yes.
the SIR, whatever they are, those compartments are essentially fractions, right? They take values of
correct.
0
but
to
there
1.
you divide people as susceptible or immune. So the immune state for an individual is either 0 or 1.
I mean, an individual can have some fraction S and some fraction I, some fraction R, such that they sum up to 1.
No. There the population is divided into susceptible individuals, infected individuals, immune individuals. So the person is either susceptible or infected or immune.
So when you're considering immunity and agent-based models, so in this case, each individual can be either S, I, or R, and there is a continuous immunity variable.
Yeah. So no, there is no S and there is no R. Only infected or uninfected is a category.
All right.
Right? Otherwise, every individual has a level of immunity, not called S or R.
Oh, okay. All right.
Okay, there is a quantitative level of immunity. Fine. So, it is possible that even in the absence of variants, a pattern of repeated waves can arise, theoretically. But should I come to this slide a little? Okay, fine. I'll go in this sequence as well. Okay. So then, what could be happening in real life? Now, this is our interpretation of real life. When you say there is an immune evading variant, the immune evasion is always partial. Okay, this is because any pathogen, whether bacterial or viral, have several proteins, several epitopes. All epitopes will not be modified with a single equation. Okay, so and with COVID as well, with SARS-CoV-2 as well, people have studied vaccine-mediated immunity to a new variant and so on. And what typically happens is, for a new variant, the titer is lower. Okay. Now, again, let me clarify. The word titer is typically used for antibodies because quantification of antibodies is easier. Quantification of other components of immunity is not that easy, not that straightforward. Okay. But conceptually, we will treat all of them as there is a level of immunity which is prone to decline in the absence of exposure, or which goes up with exposure either by vaccine or subclinical or clinical infection. Okay. So, a new variant typically has lower titer. If, let us say, the titer of 100, a very arbitrary number, for the prior variant, an immune person will show a lower titer for the new variant. Okay. Now, immunity declines. Well, now, these titers are several times higher than the minimum level of immunity required. Okay. Again, an oversimplified statement because the viral dose also matters, but if there is a level of immunity or minimum level of immunity that is required to resist infection, the titers acquired after an active infection or after vaccination are several fold greater than the minimum required, right? So, if let us say this is the minimum required, to begin with, the hosts are immune to both, but the immunity is declining. The immunity to the new variant will go below the threshold prior to the old variant. Okay, and this is a window in which the new variant has a selective advantage. The new variant does not have a selective advantage here because you are immune to both anyway. It doesn't have a selective advantage here because you are susceptible to both anyway. Okay, so the new variant gets selected in a narrow window. Right. This again is oversimplified. This window in reality will not be that sharp. Okay, but still, conceptually, there is a time in which the new variant has a selective advantage. Both prior to that and later than that, it does not have a selective advantage over the prior variant. Okay, so this is where the new variant will start invading. The mutation might have happened anywhere prior to that, but the invasion matters, and the invasion begins in this window, not immediately after mutation. Okay, yes.
You're saying that this difference in selection between the old variant and the new variant happens because of heterogeneity in the immunity of the population?
No. So it is at the population immunity. So after the peak, the population immunity will be at the maximum, right? Then the peak resides. Slowly the immunity will also start declining, right? There will be a stage in which the immunity to the new variant has gone below the threshold, but to the old variant is still enough.
Then you're assuming there is only two variants in a population, right? You know, every time there must be so many different variants, which
Correct.
might be, right?
Correct.
So what about
so extend this logic to 4 variants, 7 variants. The logic remains the same. Correct. So the immune evasion is always partial, and therefore there is a time window in which the immune evading variant will have a selective advantage. The advantage is not throughout. Okay. So this,
When any new variant is arising, so what is happening to the variants which are already there and which will be, we're assuming will be always there when any new variants are coming?
so in the absence of selection, they are subject to drift, which is random changes. They might become extinct by random changes, by chance, or they might spread a little more by chance. But there is no, see, selection is directional, whereas chance is not directional, right? So, see, mutants are happening all the time. The virus population is such a huge population that it will always be a cloud of mutants, right? The reason why we can talk about specific variants is that these variants have been selected or got fixed because of drift. Now, in viral populations, drift is also extremely complex. By drift, I mean going to fixation by chance or becoming extinct by chance. Okay. In viruses, this is also an extremely complex process. And what happens is actually a mix of selection and drift. So, out of so many mutations that are always happening, so many variants are always arising, some of them happen to get selected. Some of them happen to replace the old one, not all. Okay. Now, if you look at, now let us go by the expectation. Now, I am going to assume that no, the popular belief that new variants cause waves is what I am going to believe as well. And I will make expectations based on this. Okay, so my hypothesis is that new variants cause new waves. And if that expectation is correct, if that hypothesis is correct, what patterns do I expect in a wave? Okay, so what you expect is this. So there was a prior variant at a low level, at an endemic level, as we said. Then a new variant arises, the new variant increases, the old one either remains constant or goes to extinction. Okay, this is what we expect if the new variant is responsible for the further infections and for the surge. On the other hand, you can expect other patterns if this is not the story. And what are the other patterns? Number one, when a new wave begins, no doubt the new variant goes up, but the old variant also rises substantially and then comes down. Okay, this is a deviation from your expectation. There is another deviation from your expectation, that there is much replacement of the old variant by the new variant prior to the beginning of the wave. Okay, so the population invasion has started much before the host invasion, right? On the other hand, you might expect this deviation as well, that maybe this might be caused by the new variant, but there is another wave without any new variant coming in. Okay, so this is my expected testable prediction if variants cause the wave. Okay, these are possible deviations from the expectation. Right? Now, when we looked at the pandemic data, we looked at across variables. When we did the analysis, there were about 150 or so peaks, okay, to be analyzed. I'm not very sure about it. The number could be larger than that. It turned out that none of the peaks followed this pattern. Okay, 57% peaks followed this pattern, where the old variant also increased along with the wave. Then 73% followed this pattern and 21% followed this pattern. Don't try to add up to 100, because a single wave can show more than one pattern. Okay. So none of the waves actually showed evidence that the wave is caused by a new variant. And this is actually how the actual pictures look like. Okay, here you can see the old variant going up substantially along with the wave, then coming down. Okay, yes.
in your simulations, how many, out of all the variants you considered, how many types of decay functions did you consider? And
these are not simulations. See, this is qualitatively expected, and this is actual data.
okay.
Okay, when actual peaks were analyzed, this is the result. None of the peaks fits into this.
Okay.
Yes.
Why are we expecting the old variant to peak again?
If there is some other reason for which the infection is increasing, then even the old variant will increase. Okay, and if the old variant is also going up, it means that new variant is not sufficient reason for the wave.
So that means we are not, so in this expectation, there's no memory, no immunity.
In all of them there is.
There is, okay.
See, the new variant has an advantage for which is growing greater than faster than the old variant. But the old variant also goes up along with the wave. It means that apart from the new variant, there is some other reason why it is going up. Okay, fine. Now, another pattern that we see, yes.
Sir, in the previous slide, what is the time scale expected in the expected graphs that new both variants can come, and what is the time scale of that?
Okay, so the time is actually written here.
So
The actual dates are written here.
even in that, even in
See,
the expected,
this is a conceptual diagram. Okay, what would you expect if this is the cause? What would you expect? This is only expectation. Right? These are actual patterns which are there along with the dates.
I might sound new, but what is the difference between that A and C? I mean, conceptually, because there is only a slight difference in the time.
No, see, here the old variant has completely replaced, sorry, the new variant has completely replaced the old variant, right? And then suddenly a wave begins. There is no variant, there is no one more variant coming. Okay, the replacement has already happened.
Do you have that supportive data for that, this one, D?
Yes.
For D,
so,
do you have supportive actual
for,
data?
For 21% of the peaks,
Actual data.
yes, it was actually this.
Okay.
That a new wave arose without any new variant.
Second time, continued second time.
Yes. See, this is what is happening here. These are all actual data.
So you are plotting that there is a prior variant and Omicron and total incidence. So the total incidence is both the prior variant and the Omicron.
Correct.
It's addition.
It's just the addition of the two.
So in the third graph, in the C graph, the prevalent variant is very small compared to that Omicron. C. See, how can you expect such peak because of that thing?
Which peak?
That total incidence.
Total incidence. See,
Yeah.
see, this is because this is an addition of the two. You just add at any point, you add this, and you get this. You add red plus yellow and you get blue. It's simple addition, nothing else.
It's also not standing genetic variation.
It's also not?
Standing genetic variation. No, it could be there. It can't.
It could be there.
Yeah.
Okay. So I'm not really differentiating
Distinguishing between
mutations and standing genetic variation.
Yeah.
The thing is, the population immunity is mostly to these epitopes.
Yeah.
If there is one appears that has partially evading it,
Yeah.
will it immediately invade, or there are conditions under which it will invade? Okay, so I'm saying that it is highly conditional.
So is it the case that that was global data? Or
So that is country-wise.
that is country-wise. So can it
Country-wise.
be
So
the
these
case?
three examples come from three different countries. Now, I don't know offhand which one is which.
So can it be the case that lockdown maybe played a role? I mean, I don't.
Yeah, I'm
no
coming to
lockdown.
that.
Sorry.
I'm coming to that.
Sorry, one more question. Sorry to
Yes.
interrupt. So in your, I think in your previous graph before, you showed the waning, right? The immunity waning thing.
Yes.
Could you go back to the? Yeah, this one. No, yeah, this one. So this is at an individual level or a average of the population?
Okay. So the graph is at an individual level.
Okay.
Because the peak is sharp.
Got it.
Okay.
So
There is much synchronized immunity in the population because the peak is sharp. So many people got infected together, more or less together, and therefore their immunity levels are more or less
So
the
in
same.
the agent-based model, you did not model the heterogeneity in the arrival of the initial conditions?
In agent-based models, each individual is modeled differently. So automatically the individual variation will come.
The arrival time of the wave, is it the same for all the individuals or did you model the heterogeneity?
We don't decide the arrival time. The model decides. The wave is a result, it's not an input.
What about the decay function? Is it the same for all the?
Decay function is kept the same in the model, yes.
But for some variants, it could be that the immunity waning can happen a lot faster, a lot quicker, right?
That's possible. See, the philosophy of modeling is you start with the simplest set of assumptions. If that is not sufficient, which complexity you think is more relevant to answer your questions, bring that. Okay, if that is sufficient, stay there. If that is not sufficient, take one more level of complexity, right? So we included continuous immunity in the model.
Yeah.
Even infection level can be continuous. We kept it binary because we thought that it's okay. So,
but I was asking more in terms of the selection window.
Even the decay can be kept variable. Still, there will be a selection
variable,
window.
but if you test out two different decay rates, then
Correct.
even with the different initial conditions, the selection window would still favor the other variant, right?
See, again, you are talking about a selection window. That is the whole point. The point of the model is the selection advantage is not throughout, it is restricted to a window. Okay, that is the main point. How wide the window is, that will depend on the parameters. The parameters can be varied, no doubt about this. But it's not all time selection, there is a definite selection window. That's the point. Okay. Another interesting output of the gray immunity model. The classical SIR model typically predicts a wave like this, and with the gray immunity model or the continuous immunity model, the waves are typically dwarf by 1 or 2 orders of magnitude than the SIR model predicted wave. Okay, other parameters being more or less the same. Now, this is extremely important. Throughout the pandemic, all the waves that we have seen are dwarf. Okay, whereas SIR models predict that in order to get herd immunity, and herd immunity is the reason why it starts coming down, SIR models predict that you need 20%, 40%, 60% of the population infected. Then the herd immunity begins, right? Whereas throughout the pandemic, we have seen that it was 1% or even less than 1% or 2% of the population got infected and the wave subsided. Okay. Now, whether this dwarfness of the wave was because of continuous immunity or because of lockdowns. Okay, you can argue that, okay, the waves are dwarf because we had all these restrictions, so we just did not allow the virus to spread. Okay. Now, there is a definite testable predictions across the two. The dwarfness of the wave, is it because of lockdowns or is it because of continuous immunity? If it is because of lockdowns, then we don't expect the wave to be symmetric. If it is because of immunity, we expect the wave to be symmetric. What I mean by symmetric is, if it goes up fast, it will also come down fast. Okay, if it goes up slowly, it will come down slowly. All kinds of models show you this. Okay, that there is a correlation between the rate at which it goes up and the rate at which it comes down. By symmetry, it is not always bilaterally symmetrical. Okay, but symmetrical in the sense that if it goes up fast, it also comes down fast. Okay, so this is again in a model where you see that the one that goes up fast comes down fast. The one that goes up slowly also comes down slowly. This is what you expect as an effect of immunity. If it is because of lockdown, then it depends on the stringency of restrictions. It depends upon the population density. It depends upon whether people live in a congested environment or an open environment. Okay, and therefore, there is no reason why it should be symmetric. In fact, if it is symmetric, it is a rare coincidence that it is symmetric. And what you see throughout the pandemic is extremely good level of symmetry. The rate of going up and the rate of coming down are very tightly correlated. Okay, and therefore, it is unlikely to be the restrictions, the lockdown that made the waves dwarf. It must be the immunity effect that is working there. Okay, now let us come back to this picture. Okay, so in all countries now we have data of how the different waves are, how the different variants are coming and going. Okay, and on a time match scale, the waves are plotted. Okay, now since so many variants have been arising, it is quite likely that a peak coincides with a variant by chance. This is the basic principle of science, that you first make a null hypothesis of chance, and only by rejecting the null hypothesis, you would say that there is an association. Okay, so what is the chance by which a peak coincides with a variant by chance? Believe me, people have been talking about the variants causing waves all the time. Nobody had done this analysis. Not a single paper on whether the association is by chance or not. Now, some of my co-workers have started analyzing it. We haven't yet completed the analysis, but you can imagine when so many variants are succeeding each other, there is a good chance that a randomly placed peak lies along with the rise of a variant. Okay, so we are actually doing this exercise. I will show you only a preliminary result. Now, what is this? This is the frequency distribution of the rate at which variants increased and invaded. Okay? Now, what would I expect? If the variants are highly infectious and therefore they cause the new waves, the variants associated with waves the variants associated with waves should be more on the right-hand side. Do you get this point? Okay, if more infectious variants are responsible for the waves, then in this frequency distribution, variants associated with waves should show greater growth rates. They should be on the right-hand side. Okay, look at what is the actual distribution of all the variant growth rates and variants associated with the distribution is not very different. Okay, only slightly different. Now, this analysis is not yet complete, but I have started believing that the waves can be placed independently along that line, and then the picture that you get is something like this. Okay, which means that there is no real evidence that variants and waves are associated. Forget about causation, even association is doubtful. Association is not beyond chance. Okay, so there is no reason to believe that variants cause waves. Okay. The second interesting, yes. Huh.
when I analyze the data, what we collect, I do not agree with that because clearly we see the variants which are increasing, the growth rate is related to those variants. So I'm not able to understand why would you come to that conclusion.
Did you look at all waves and all variants?
Yes.
Then show the graphs.
I will. If you are here on 9th of July, I will. No, the reason I say so because see, it's a lot of clinical data rely on the number of cases increasing and everything.
Yes.
And all the data for the sequencing, for genomics, for the clinical data, it depended on how many people are sequenced at a given time.
Correct.
Right? So it's kind of a biased one. I do not work on clinical data. I do a lot of wastewater sequencing, which is like samples are collected every week and then we are sequencing, irrespective of whatever the viral load is. So when the viral load is increasing and decreasing, we have a very mapped data for that. We're also looking at different mutations associated with different variants, and we see this growth rate, how that is changing. That's one part. But B, if it is just by chance, then why only one variant becomes dominant across all over the world at the same time? Do you have something to say? So I'm just a bit baffled by the whole logic here.
Correct.
That's why. Yeah.
So what I'm saying is the waves and variants are unrelated to each other. This does not mean that it is by chance. See, selection happens at so many different levels. There is selection within cell. There is selection for transport within a body from one cell to another. There is selection for survival outside the host. Okay, not all of them are related to infectiousness or virulence. So there can be selection at various levels which we have no idea about. And therefore, the succession of variants would have some selection there. But that selection did not be related to virulence or infectivity. Yes.
So the variants are the actual products of selections, right?
Succession, yes. So selection.
No, the products of selection. The variants are the products of selection, right?
Correct.
Yeah. If that in case, so these variants are what you're saying means I don't know. So what I am understanding is the variants are supporting the emergence of the previous one. No.
The variants have outcompeted the previous one
Yeah.
for some reason. The reason we do not know as yet.
How come variants are the product of selection? I don't understand that.
Selections is where the wild type will get adapted to the environment, right?
No, I think selection will act on individual variants.
Correct.
Right.
So,
No, not random mutation. Yeah.
correct. But that mutant has to invade the prior variant, for which it needs some kind of advantage.
But not all variants have advantages. There's a
How
lot
do
of
we know?
neutral.
How do we know? Because the only thing that we look at is the epidemiological picture. We don't look at what happens at the cell level, what happens at the host blood level, what happens at the lung level. That we do not know.
But from whatever we know of other studies on viruses and things like that, the distribution of fitness effects, we expect that most variants, most mutations would be detrimental and very, very few will be advantageous, many will be neutral. So that's the expectation at least, unless COVID-2 is very different or something like that.
I suspect that it could as well be very different.
So variants are not the product of selection, but selection is a parameter where we can find out the variant. So even there is no selection, it does not mean that there is no emergence of variant.
See, mutations will keep on happening all the time, right? When you say that a particular variant has replaced the old variant, there are only two possibilities, either drift or selection. There is nothing else that can do this. Okay. Huh.
Drift in the sense, like some, unexplained mechanisms that you are referring to?
Drift is purely by chance. Purely by chance. Okay. And by chance, they can increase, or by chance, they can become extinct as well.
Sampling. Or sampling.
Okay. So, should we talk about another issue, or you have, yeah. No, or you have sufficient to debate on this issue itself. Okay, fine. So another issue has been about virulence, and it is not about this pandemic. The issue of evolution of virulence has been discussed over quite a wide variety of bacterial, viral pathogens, of animals, of humans, and so on. Okay. Now, in the right from 1980s, there has been substantial literature on why some are more virulent, some viruses are more virulent than others, both across species as well as within variants of the same virus, why some are more virulent than the other. This has a long history. Okay. Now, what you see here is during the pandemic, the case fatality ratio declined right from the beginning, and it has been more or less monotonically declining. The total number of deaths might have gone up, okay, but the case fatality rate has monotonically declined. And it started decline much before vaccines became available. Okay. So this is what the history of the debate of evolution of virulence has been. The classical concept, which has been there for the long time, is that pathogens evolve towards reduced virulence, because if they destroy their host, they will destroy themselves. Okay, so pathogen evolution should lead to reduced virulence. But then people found that this is not the case with every virus. For example, smallpox virus has been with us for 3,000 years, no signs with decrease in virulence until it became extinct, but there was no signs of decrease in virulence. So this is not what happens all the time. Okay, sometimes the virulence decreases, at other times it doesn't. So then the question is, what are the conditions under which there is evolution of reduced virulence? What are the conditions under which virulence either remains the same or even increase? Okay, and then there has been a lot of development on that. There is substantial literature. I just quoted a few, but I would encourage you to go, an extremely important piece to read, extremely important volume to read actually. Okay. And very important empirical study is about the myxoma virus. The myxoma virus is a virus of rabbits that was introduced into rabbits in Australia after a couple of decades that rabbits were introduced to Australia. Rabbits are not Australian animals, okay, but Europeans introduced them to Australia as a game animal. They wanted something to hunt, something to take a pleasure of hunting, and so they introduced rabbits there. Very soon, not having any predators, rabbits spread like anything. They became a menace, and completely uncontrollable menace. So as a experiment in biocontrol, they introduce myxoma virus there. Oh, this works automatically, is it? Okay, I'm not operating. Okay, but fine, I can come back anyway. So what happened is, initially the myxoma virus killed almost 99% of the population. Some survived. But then two things happened. Those survived were immune, their immunity levels to myxoma virus were high, but the myxoma virus also lost its virulence. Okay, the two are separate processes. In rabbits, it is possible to separate the two because you can use it as an experimental animal. In humans, we cannot segregate the two because there is no experiment involved, right? But in rabbits, it was very clearly shown that as the rabbits became immune, the virus also lost its virulence. Okay, the reason given there for losing virulence is something like this. Let us say there were two types arising by mutation or prior variant, whatever it is. Let us say B is more virulent than A. But there the meaning of virulence is very specific. I will come back to this. The virulent one is supposed to replicate faster. Okay, and there is a question mark on this. I will come to that. So here their argument was that within a body, B will replicate more than A. But across rabbits, if you have some which are infected by A and some infected by B, then the ones infected by, what is this? The ones infected by the more virulent one will die. The less virulent one will allow the animal to survive and move around spreading the virus. Okay, and that is how the less virulent virus will spread in the population, the more virulent virus will kill itself. And that is how evolutionarily the variance will keep on going down. Okay, now this is both well modeled as well as empirically demonstrated, and empirically demonstration was possible because experiments were possible.
Excuse me.
Yes.
So is there any proof for that to have two strains instead of one?
Well,
is that because of the presence of one less virulent and
for
more
myxoma
virulent,
case,
or otherwise,
for myxomatosis,
virulent virus itself is reduced its virulence during the period of time.
See, evolution is always new mutants arise which are different in character than the ancestor, than the parents. So
you explain the concept by
and
taking
B,
two strains.
I showed A and B. They could have arisen by mutation in the population itself, or they might already be there in the virus that came in.
So as you said in the beginning, that pathogen reduces its virulence by time. But here, the conclusion, it is very difficult to arrive using two different strains. Rather, instead of two, you select one. So thereby, you can
see even if you start with one, mutations will arise, which is a different phenotype. So that is A and B. Whether initially there was only one or not is a different question, right? But in evolution, mutations are necessary, right? Please understand, I am not saying mutations are not necessary. I am saying mutations are not limiting. The two arguments are completely different. Mutations are absolutely necessary for anything in evolution to happen. Okay. Okay. Then there are other examples of virulence declining. Leprosy is an extremely good example. Okay, leprosy has not been eradicated. Leprosy is still there, but the kind of lepers that we used to see in my childhood are just not seen. Today's generation just does not know lepers. Okay, they used to be around so many. There are two things happened. Good drugs were made available, but number two, there is evidence that leprosy lost its virulence. Mycobacterium leprae lost its virulence. Okay. Now, this is something that has been happening with the COVID pandemic too, that the case fatality ratio has been declining all the time. But why is it? Since we do not have an experimental system, it might be declining just because you detect more asymptomatic cases, or it is because the age class of patients has been changed. So there are so many alternative explanations, alternative hypotheses to explain the declining CFR. And we systematically analyzed all of them, and by eliminating others, we could show that yes, the virulence is declining. The others are not sufficient to explain. I'm just showing one example here, and that is whether your treatment became more effective and therefore the mortality declined. It is not so, because this is all prior to vaccination. Okay, all this data is prior to vaccination. The percent hospitalized declined, but among hospitalized, the death did not decline. Okay, which means that the severity came down, but the treatment did not improve. There is no sign that the treatment actually improved. the need for hospitalization itself came down. Okay, so now there are the last two which remain. So today, today we know that it is still there around, but hardly anyone cares, hardly anyone bothers, because the mortality has come down almost equal to any other influenza or any other common respiratory disease. Is it because of vaccination, or is it because the virulence itself has come down? Okay, after having eliminated the other possibilities, right? Whether it is in effect of vaccination, or whether it is, as we said, in myxomatosis, you have experimental animals, so there segregating this was possible. In human data, segregating these two is tough, but it is possible in one particular case, and that is Australia and New Zealand. Okay, Australia and New Zealand had some initial cases. They were in a few hundreds, okay, not in thousands, but there were. Then, for a long time, both Australia and New Zealand kept the virus away from their countries effectively. And then they couldn't do it at a later stage. And then at a later stage, the incidence was in fact worse than India, okay, in terms of cases per million. In fact, in terms of cases per million, India is at a very low rank as compared to, so even including or correcting for the underreporting. Okay, so these were very badly affected. But if you take the case fatality rate in the beginning and after the gap, there is a 30 to 35-fold difference in the case fatality rate. Okay, remember, 30 to 35-fold difference in the case fatality rate. During this long period, there was no natural infection. So there is no question of natural immunity. Vaccination had begun and saturated at about 80%. Right? The rest of the 20% refused to get vaccinated. So now, if the decline in mortality is because of vaccination, then we can make two sets of assumptions. Number one, that the vaccination did not cause a difference in incidence. Okay, but because of vaccination, the mortality came down to 0. The other extreme example is that vaccinated individual did not get infected at all. So whosoever got infected was among the unvaccinated. Okay, if the second case is true, there is no reason why mortality should have come down. You are saying only the unvaccinated got infected, but even in them the mortality came down. So that is certainly not an effect of vaccination. If the first one is true, then only 5-fold reduction is explained by vaccination, because 80% were vaccinated. From 100 you came down to 20. So 5-fold is the maximum reduction possible by vaccination. In reality, there is a 35-fold. Okay, which means that in these two countries, we can certainly say that vaccination is not at least solely responsible for the reduction in mortality, and there is something unexplained by vaccination. So what could that be? Okay, so again, let me skip a few things, but show you the important things. So many papers show that there is no or very poor correlation between the viral load and severity. Okay, now this is extremely important. Why should be a pathogen, or what would a pathogen gain by being virulent? The only thing it can gain is rapid replication in the body. Okay, so if virulence is related, tightly correlated to replication within the body, then there is a selective advantage in being virulent, right? If the two are uncorrelated, there is no advantage in being virulent. And if there is no advantage in being virulent, then even a small disadvantage will lead to loss of virulence, right? In COVID data, we see across so many studies that there is very poor correlation between viral load and severity of infection or severity of symptoms. The other thing is, one of the peculiarities of SARS-CoV-2, which is not shared by other coronaviruses, is it attacks blood vessels. Okay, and many of the fatal effects were because of blood vessel attack, not because of lung infection alone. Now the thing is, the viruses that attack the blood vessel have no escape route. The viruses that are transmitted are the viruses growing in the upper respiratory tract, right? So there is no advantage to the virus in attacking blood vessels, correct? So by being virulent, by being fatal, the virus does not get any advantage. Now, there is a slight disadvantage here because you quarantine the infected individuals, right? So the virus has less chance to spread. A virus which is milder will allow the host to roam around and spread it. That slight advantage is enough because there is no advantage in being virulent. A slight disadvantage works, and therefore throughout the virus has been monotonically reducing its virulence. But this is a politically incorrect statement. Okay, and so many journals actually refuse to publish that the virus is becoming mild. And there have been many other arguments without any data, without any evidence that, oh, suddenly new, more virulent virus has arisen. There is no data shows that. Okay, so one last point. The vaccines were disappointing. Okay, I'm not saying they were more dangerous than, so they were useful, no doubt, but not as much as expected. We expected that when the smallpox vaccine came and the entire population was vaccinated, smallpox just disappeared. The same thing is happening with polio. Already happened in many countries, but now they still want to keep a margin and therefore vaccination still continues. So smallpox and polio were wiped off by the vaccination. That did not happen here. In fact, even the vaccinated individuals became susceptible and were again infected after 4 months, 6 months, and so on. So the vaccine was disappointing. The question is why? Why smallpox, polio are so different than this? There is an evolutionary logic. It is speculative. Right now it is not being tested, but this is my logic. Immunity is costly. Okay? Immunity is very costly, not only for the cells that are devoted to memory, the cells that are devoted to immune system. Inflammatory response damages your own tissues. Okay, and therefore immunity is a costly affair. Therefore, there is no point in giving immune response to a pathogen which doesn't cause much harm. Okay, our response to common cold is very mild, not long lasting. For long lasting response, you need more investment, greater investment. Okay, and for milder viruses, that investment is completely unnecessary. So our body has evolved over thousands and millions years of association with viruses. Our body has evolved this distinction that for potentially dangerous viruses, you invest more in immunity, like pox viruses. For potentially or rarely virulent viruses, such as coronaviruses, don't invest much in immunity because your cost of investment will be much greater than the cost of infection. Okay, so the body doesn't give full-fledged immune response to generally milder viruses. See, among coronaviruses, this is an outlier. No other coronavirus is known to be this virulent. So our body doesn't invest. Now, if that is the case, that also gives a tentative answer to another question. For smallpox or for polio, we don't know any variants. The vaccine worked, it eliminated, no variants arose, there was no immune invasion, no immune evasion. Okay, why was that? The simple reason is because the immunity is long lasting, the selection window never arises. Okay, so not that these viruses don't mutate. All viruses mutate. Show me a virus that don't mutate, I will give you 10 Nobel Prizes. This is just not possible. All viruses mutate, but since the selection window is absent, new viruses don't get selected, new variants don't get selected. So for smallpox or polio and viruses like that, where the body invests a lot and the immunity is long term, the selection window does not arise and therefore new variants do not arise. Okay. I'll just make a simple comment on this and stop. I will just say that the evolutionary dynamics of spillover or jumping across hosts is something that I say with 100% confidence that I don't understand, and I say with 99% confidence that nobody in the world understands. Okay, we just know that this happens, right? But as I interpreted the evolution of variants, evolution for spillovers still does not have a good, logical, sound, mathematically working argument. We just know that this happens. Okay, but see, there are so many things that we haven't just thought of. For example, see, this possibility has been there all the time, and jumping host spillover has been there all the time. There are so many examples from written history, whatever. So our bodies also must have evolved some strategies against it, and they have. During my PhD, which was 35 years ago, and none of these talks were there, I asked this question: how do we differentially treat wild animals from which we are more likely to get infections and from which we are less likely to get infections? Okay, so for example, human zoonotic infections have, I'm not only talking about viruses. I was more working on helminths and protozoans. Okay, so my question was slightly different that time. So we could show with both experiments, simple psychology experiments, simple human behavior experiments, that the animals from which you are more likely to get zoonotic infections are avoided by humans intrinsically. Okay, I used to do fecal analysis for parasites. I used to take fecal samples, and many of our tribal assistants and field guides also used to do that. They would easily collect deer sample, they would easily collect elephant sample. They were extremely reluctant in collecting carnivore samples. Okay, and carnivores have much greater possibility of zoonotic infections, at least in terms of protozoan and helminths, than herbivores, particularly the hoofed animals. Okay, there are hardly any zoonotic infections that we get from hoofed animals. On the other hand, animals from which you are more likely to get zoonotic infections are hated and avoided by most people. Okay, this could be either a biologically evolved or a culturally evolved response. Yes, but there are exceptions. There are exceptions. There are.
I love bats. Yeah.
So,
I mean, guano extraction is a very, it's in a lot of places people collect guano for fertilizers.
Correct, because
and
that gives money. That gives money. Okay. And
so if you pay your field assistant to collect carnivore samples, they should collect carnivore samples also. No,
So I made them collect all the samples that were necessary. Okay, but by themselves they were reluctant to collect samples from some species. They did not mind other species, right? There are exceptions, and these exceptions are important. So there is a community called Vaidus in Maharashtra who used to hunt wild cats and eat them, right? Now this is generally avoided, but Vaidus did that. Now it is necessary to study. Now they are vanishing. These communities are vanishing, or their culture of catching. I have seen it, but I have seen it 20 years ago. I do not know whether it still exists. I have seen their traps by which they catch wildcats. Okay, but they used to eat wildcats. It is necessary to study these communities. Okay, because they might have exposed to the potential zoonotic viruses and so on. Okay. Fine, so let me stop. There are so many possibilities and so many things I like to work with.
Thank you so much. Other questions? Yeah.
Sir, it is just general question.
Yes.
Can we correlate, compared to wild animals, most of them are adapted to the pathogens. So act as a reservoirs, vectors, without showing symptoms or without, thereby, as you said, co-evolution. So can we correlate with human civilization? So where we are avoiding the interaction with all types of pathogens. So we are not permitting the pathogen to interact continuously by using medicine, vaccinations, so
Yes.
many things. Can we correlate this? Hygienic environment.
Yes. So there have been published papers. For example, I mentioned New Zealand and Australia. Okay. So they kept very stringent restrictions and they kept the COVID virus away. During that 1 year, several milder viruses are reported to increase their severity. Okay, this is something, and there are published reports of this. There is a debate. Okay, not everyone has agreed, but so this is reported. In 1997, we reported in Lancet that in Pune, families that use all the time, all seasons, use water filters or only drink boiling water and so on, are more susceptible to gastrointestinal infections. Lancet published this 25 years ago. Okay, so so that kind of immunity does make sense. And what I feel, this is again speculation, no evidence in hand, that over the last few generations, we are getting increasingly isolated from wild animals. And that is what makes us more susceptible to zoonotic infections. Yes.
Okay. So I actually have 3 questions. So the first is that, is it common to see this lack of correlation between this virulence and transmission that you were showing for SARS-CoV? Because that thought that was...
For SARS-CoV, 3-4 papers seem to agree on this.
Right, but is there any other pathogen that we see this similar kind of non-correlation?
Exactly opposite is the case of smallpox. Correct. In smallpox, virulence and viral load are tightly correlated, and therefore smallpox never reduced its virulence.
Even for TB, it is?
Yes.
Yeah, okay.
Yes.
The second is that I was reading another paper that was talking about this Muller's ratchet as a potential mechanism why these asexual...
I know what paper you are talking about. Yes.
This RNA viruses which have very high mutation rates, they just like their virulence just falls off because of like mutation load.
Yeah. So I know which paper you are talking about. So this paper was very interesting because they showed that the virulence is declining prior to vaccination. This was one of the very early papers, right? And but that Muller's ratchet argument doesn't hold because Muller's ratchet is for larger genomes. It doesn't work for smaller genomes. Virus is too small for Muller's ratchet.
And the last one, in your, you said that it is probably not mutation limited, but we do find that these RNA viruses which have very high mutation rates are the ones that are most likely to spill over. So why is that?
About spillover, I really don't know. Okay. The only possibility I can think of is spillover, like cancer, needs a combination of mutations, which is extremely rare. In that case, mutation will become a limiting factor.
Also, I don't know, I was wondering whether you think that spillover actually does happen often, but a disease does not happen as often. Like there's not the ability to replicate in that host is not there.
Well, I suspect replication also would be very common, more common than what we think, but further human-to-human transmission may not happen. Mostly it doesn't happen.
In one of your graphs where the death rate was naturally decreasing until the vaccination, at vaccination after that, why did it flatten? It should have dropped further down, like even steeper?
I don't think that is very significant. So more or less there is a downward trend. There will be some slope changes along that.
So it was a natural slope change. Vaccination did not affect it because it exactly at that point it flattens.
Yes. That is noticeable, no doubt. But if that is the trend, I don't know why.
Thank you so much. Lots to think about, and yeah, thank you.